Basic components of transdermal drug delivery system. It contains polymer matrix, drug permeation enhancers, pressure sensitive adhesive, and backing laminates, release liner, and other excipients like a plasticizer and solvent. In these components of basic components of transdermal drug delivery system, the polymer matrix control the release of drug from the device. The polymer matrix can be prepared by the dispersion of drug in liquid or solid state synthetic polymer base. These polymers are used in transdermal drug delivery system, should have good stability and compatibility with the drug and other components of the system, and it provides the effective release of drug throughout the device with safe status. For example, cellulose, gelatin, shellac, waxes, gums, and natural rubber and chitosan. Next one is permeation enhancer, which enhance the permeation in transdermal drug delivery system. In these permeation enhancers, these compounds are useful to increase the permeability of stratum corneum by interacting with the structural components of stratum corneum. Proteins or lipids attached to the high therapeutic levels of the drug. They can change the protein package. So that's why it is also called as protein enhancement. Pressure sensitive adhesives. These PSA affix the transdermal drug delivery system firmly to the skin. It should adhere means increase the these adhere with not more than applied finger pressure. Adhesives must be skin compatible, causing minimal irritation and sensitization, and these are removable without inflicting physical trauma. The pharmacological effect of these pressure sensitive and adhesives are without losing their adhesive properties and skin tolerability. Another one is backing laminate. In these backing materials must be flexible while processing of the good tensile strength. These backing laminate commonly used materials are polyesters, elastomers, pigmented materials, etc. Another one is release liners. During storage, the patch is covered by the protective liner that is removed and discharged immediately before the application of the patch to the skin. Like when there is any edema is formed, then that's time we can use the patch that removes the swelling. So that's why these are used. And other excipients like solvents such as chloroform, methanol, acetone, isopropanol, dichloromethane, these are used to prepare the drug reservoir. These are the some basic components to use in the transdermal drug delivery system, and evaluation methods of transdermal drug delivery system. What are the evaluation methods? Here, interaction studies are there. Some physicochemical evaluation methods are seen in transdermal drug delivery system. They are interaction studies. The drug and excipients must be compatible with one another product. It means, for example, in marriage life also compatibility is main thing to be sustained. Okay, that's why here the interaction studies is the main important topic, and then thickness of the patch. The thickness of the drug prepared patch is measured by the using digital micrometer. At different point of patch, the determination of thickness and standard deviation will be changed. Okay, and weight uniformity also a evaluation process. In this weight uniformity, the patches are dried at 60 degrees Celsius for 4 hours before testing. After that testing, the specified area is used to weigh that patches, and then the folding endurance will be started. Yes, this folding endurance is a specific area of strip which is cut and repeatedly folded at the same place till it broke. That patch until the patch will be broken, this process will be continued. And percentage moisture content, the percentage also checked by a device to remove the moisture content in that patch. That's for example, if is there any moisture in that patch, it doesn't work on the skin. So that's why to remove that moisture, the some devices are used to remove the moisture content in that patches. And then drug content, a specified area of patch to be dissolved in a suitable solvent in specific volume. Then the solution is filtered through the filter medium and then analyzed the drug content with suitable method like UV, HPLC techniques. Then we have taken the average of the 3 different samples. Then we check the content uniformity test. In this content uniformity test, the 10 patches are selected and they are determined individually. Out of 10, 9 will be having 85 to 100% specified value. That time only that content uniformity test will be passed. Otherwise it will fails. Okay. And then uniformity of dosage unit test. In this uniformity of dosage unit test, the accurately weighed portion of the patch is to cut into small pieces and transferred into specific volumetric flask, and then the patches will dissolved into the solvent and sonicate for the complete extraction of drug for that patch, and resulting solution was allowed to settle for about 1 hour. After that, the suitable diluted desired concentration will be came out with the suitable solvent. After this process, the polaroscopic examination will be done. In this polaroscopic examination test, a specific surface area of piece kept on the object slide of polaroscopic and observe for the drug crystals to distinguish the drug in present crystalline form, which form, amorphous or crystalline. To determine the form of the patch, this process will be performed. And then after shear adhesion test, in this test the cohesive strength and polymer adhesiveness will be measured by a device. It can be influenced by the molecular weight, and the degree of cross-linking and composition of polymer types are added to increase the adhesiveness. This is about the methods of evaluation process in transdermal drug delivery system. After that, we are going into the in vitro evaluation of transdermal delivery system. There are two types of evaluation test. We already discussed about that, in vitro, in vivo. Now we are discussing about the in vitro evaluation. In these in vitro drug release studies, the paddle over disc method is used to release the drug from the prepared patches. Dry films of thickness are cut into definite shape and weighed that and then fixed to a glass plate, and that glass plate will be placed in a 500 ml of dissolution medium. After that, the apparatus will be equilibrated. The pedal was set at a distance mm, accurately 2.5 centimeters from the glass plate, and the speed of that paddle is 50 revolution per minute, and then the appropriate time intervals will be noted for up to 24 hours, and then we can analyze that patch under the UV spectrophotometer. This experiment is performed to in the This experiment is performed in triplicate, and mean value can be calculated. After that, in vitro skin permeation studies will be conducted. In these in vitro skin permeation studies, this is used by a diffusion cell. Full thickness abdominal skin of a male Weistar rat is weighed at 2. We need a male rat, which is nothing but Weistar rat. That weight must be having 250 grams, and hair from the abdominal region is removed carefully by using an electric clipper, and then that skin will be cleaned thoroughly, and then we have to remove the tissue of our blood cells. In that place we have to cut that place and equilibrated for an hour in dissolution medium. Before starting the experiment, we placed a magnetic stirrer with a small magnetic needle for uniform distribution of the diffusant. The temperature of the cell was maintained 32 degrees using a thermostatically controlled heater. After isolation of the rat skin piece, the compartments will be made by using the clipper. After sample volume of the definite volume is to be removed from the receptor compartment at regular intervals, and equal volume of the fresh medium is to be replaced. Samples are filtered through the filter medium and analyzed spectrophotometrically or by using HPLC method. The flux can be determined directly as the slope of curve between the steady state values of the amount of the drug within 1 hour, as permeability coefficients will be deduced by dividing the flux in the initial drug load. This is the in vitro evaluation. Means in this in vitro evaluation, rat skin piece is taken outwardly, and then we have to perform the experiment. In vivo evaluation, the animal species are used for evaluating transdermal drug delivery system in mouse, hairless rat, hairless dog, hairless rhesus, hairless monkeys, hairless rabbits, or a guinea pigs, etc. We need a hairless animal for the experiment, and then we have to perform that experiment in vivo, means inside the body. After that animal models, we have to perform the human models means like volunteers, clinical trials. For example, clinical trials conducted that determines the safety in volunteers, and after that in phase 2 process it determines the safety and effectiveness in patients. In phase 3 trials, the large number of patient populations will be examined by these samples. After phase 4 trials, the post-marketing surveillance will be done. This is the in vivo evaluation of the transdermal drug delivery system. Okay, another study is also there extra, that is skin irritation study. This skin irritation study is performed on the healthy rabbits, which is around average weight is 1.5 kgs. The dorsal surface is 50 centimeters of the rabbit is to be cleaned and remove the hair from the clean dorsal surface by shaving or any removing of the hair by using the rectified spirit, and the representative formulations can be applied over the skin, and the patch is removed after 24 hours, and skin is to be observed, and this classified into 5 grades on the basis of the skin injury. This is the skin irritation study. Okay, this is about the evaluation methods and components, basic components which are used in the transdermal drug delivery system. Thank you for listening, thank you for watching. Please like, share and subscribe to my channel. Thank you very much for listening my class.